Hello everyone and welcome back to Biology with Mrs. Evans. Today we are going to be looking at how proteins are made. So we finished Unit 6, which is all about nucleic acids. So we briefly talked about the different types of RNA and how those are used in the process of making proteins. But now we're going to go a little bit further in detail regarding how proteins are made. So messenger RNA, remember messenger RNA is single-stranded and it is a copy basically of DNA. As you see here on the picture, you have DNA serves as the template, and then instead of DNA polymerase reading the exposed bases, RNA polymerase is going to read those, and it's just like DNA replication. It's going to bring over the corresponding bases. So you see adenine is exposed, so it's going to bring over uracil, another adenine, uracil, another adenine, uracil, and then cytosine is going to bring over guanine, another guanine, and then you can see it's going to bring over cytosine for that. So RNA polymerase is going to read the exposed DNA strand and use that to make messenger RNA. Transfer RNA, remember, is a type of RNA that brings over amino acids to the ribosome based on the reading of messenger RNA, and I'll go over that again in a moment. Ribosomal RNA, remember, ribosomes are made up of ribosomal RNA, and it wasn't until recent that scientists learned the role that it plays, and they're still finding out more about it, so more will probably be known in the future. But right now we know that ribosomal RNA helps hold messenger RNA to the ribosome. So the big idea about protein synthesis is that it's made, basically has two steps. You have transcription, which is DNA is used to make messenger RNA, and then you have translation, which is messenger RNA is going to be used to make a protein. So it all starts with DNA and it ends with basically a protein being made. So remember, messenger RNA carries the information from the nucleus, all right, because remember, DNA cannot leave the cell because it's double-stranded. It's too big to fit through. Messenger RNA is single-stranded, so it can carry that message to the ribosome. And then messenger RNA is going to basically be the recipe for how the protein is being made, and I'll get into that in a moment. And then transfer RNA, remember, to me, I remember it because they look like little trumpets. All right, this is a transfer RNA molecule. They bring over amino acids based on the codes that are on messenger RNA, and I'll go over how that works in a moment. So transcription, you heard me say this in Unit 6. Transcription is the process that basically creates messenger RNA. DNA is going to be used as a template to make messenger RNA. So again, helicase is going to come in. It's going to break the hydrogen bonds that hold the two strands of DNA together. Then instead of DNA polymerase reading it, it's now going to be RNA polymerase, and it's going to make a strand of messenger RNA, just like how DNA polymerase makes a complementary strand of DNA. So RNA polymerase will read the exposed strand and bring over the corresponding bases. So let's review. So here's my exposed DNA strand that I have here on the screen. So remember, A would bond with U this time because remember, RNA does not have thymine. So A, again, bonds with U, C bonds with G, G bonds with C, T bonds with A bonds with U, G, C, A. So this would be my messenger RNA strand, and it would now leave, once it's made, and exit the nucleus and arrive at the ribosome, where the ribosome would then read it and use it to make a protein. So a good portion of DNA is what we call basically junk DNA, and that's because it doesn't, as of right now, scientists don't know what it codes for. We believe it is just junk DNA nonsense. So before it can be used to make a protein, it's going to be edited. It's going to remove all that junk that doesn't code for anything, so that when it arrives at the ribosome, then it only has the specific information for how to make a protein for that particular protein encoded. So what happens is there's going to be some editing. And this picture here, again, is just showing you how messenger RNA is basically created. So helicase will break the double helix as you see, and then messenger RNA is going to be made from the DNA, because RNA polymerase will read the exposed strands. And you can see that it follows basically the base pair rule that we talked about. And the difference is, obviously, uracil is going to be used in place of thymine, because messenger RNA does not have thymine. So once it's created, messenger RNA is going to be edited. So the pieces that basically do not code for anything are going to be cut out. And those are what we call basically introns. So introns are basically the junk DNA, and they're going to be cut out. And that means basically they're in size, which means to cut out, so that's why we call them introns. And then all of the pieces that basically do code for something are going to be basically spliced back together, kind of glued back together. And the pieces that do leave basically the nucleus and are going to be read by the ribosome are what we call exons. And the way to remember that, exons exit the nucleus and are going to be expressed. So those are what are going to be used by the ribosome to make a protein. So this is basically a strand of messenger RNA, and the way that the ribosome reads it is it reads it in groups of 3. So this is 1, this is 2, and this is 3. Each 3 base sequence found on messenger RNA is called a codon. It's called a codon because that 3 base sequence is going to code for a specific amino acid. Now, the way that the messenger RNA reads the messenger RNA is it's looking for a start codon, and that start codon is this right here. And this is something that you are going to need to know. AUG codes for a specific amino acid. It's called methionine, and it's also going to tell the ribosome, hey, this is where you need to start making the protein. And it's going to read it again in 3 base chunks to know which amino acid to bring. So now transfer RNA, remember I said it's going to bring over the amino acid. Remember we talked about it in class. Transfer RNA has the anti-codon. It's a 3 base sequence that's found on transfer RNA that is going to correspond to the messenger RNA. So the anti-codon for AUG would be UAC, and that tells it basically to bring over methionine. And then this one would be anti-codon would be AG, and that codes for a specific amino acid, and then U would also bring over 1. So transcription is the process of making messenger RNA from DNA, and we're going to look at how translation takes place in the next video. Thanks. Have a great day. Bye.